Great, and I think we are live. Wonderful. So, hello everyone, and welcome to another worldwide Neuro talk in our Sussex Vision series. My name is Nora, and I'll be hosting our talk today. At first, I would like to thank again all of the worldwide Neuro organizers for coming up with this whole idea and making it possible for all of us to enjoy such an enormous variety of talks and expertise during this spring and summer. It has been really wonderful to hear all these different kind of talks. Next, I would like to remind our audience that you can send your questions directly in the chat on the YouTube, and we will collect the questions and ask the most popular ones right after Todd's and Emily's talk. So today we have pleasure to hear a joint talk by Todd Thiele from University of Toronto and Emily Cooper from University of California, Berkeley. Todd did his PhD in Sean Lottery's lab at the University of Oregon, studying the neural circuitry of chemotaxis behavior in C. elegans. During this time, he also got interested in zebrafish as a model animal, and he did a postdoc with Herwig Bayer investigating motor control circuits in zebrafish. Now, Todd's own group is studying zebrafish neural circuits involved in sensomotor integration. Emily did her PhD at University of California, Berkeley with Martin Banks on depth perception in real and picture environments. After getting her PhD, she conducted postdoctoral research at Stanford University and then joined the faculty at Dartmouth College in 2015. Emily moved back to Berkeley in 2018, where her work examines the principles underlying biological vision with applications to both basic and translational questions. So today, the topic, our first joint talk, is understanding the visual demands of underwater habitats for aquatic animals used in neuroscience research. So Todd and Emily, you're welcome to start.
Great. Well, I want to thank Nora and Tom for the invitation and the organizers of the Worldwide Neuro series for this exciting opportunity to talk about this project. And again, this is a joint talk between myself and Emily Cooper, and we're going to be talking about understanding the visual demands of underwater habitats for aquatic animals used in neuroscience research. So part one of the talk I will give, and this will be a project overview. We'll talk about some of the field research we have done and some engineering efforts related to future aspects of the project. And in part two, Emily will discuss an analysis of natural scene statistics from zebrafish habitats. Okay, kind of a big picture slide here. So when trying to understand how a perceptual system works, taking a neuroethological approach is often quite useful. And so what you want to understand are a couple of aspects, particularly what behaviors that perceptual system helps to shape and drive, and also to have an intimate understanding of the environment in which that perceptual system evolved. So when studying vision, this of course involves understanding natural scenes. And so the theoretical space of visual imagery is effectively infinite, but the images we see in the natural world have many common features. Statistical regularities in natural visual signals can be exploited by the visual system to efficiently and accurately perceive the world. So here we just have a terrestrial and an aquatic scene, gorillas in a jungle and fish in a coral reef environment. Now, the ultimate goal for our project is to understand the visual processing of optic flow in natural scenes. And so optic flow can be produced either by self-motion or motion induced by environmental factors such as wind and current. And during self-motion, optic flow can be quite useful for an animal to understand how fast they're going, how well they're navigating in an environment. And for fish and also animals that fly, external factors such as wind and current are able to move animals such as these quite strongly in their environment. And so these animals have quite effective behaviors for stabilizing themselves within dynamic environments. And so I'll move away from natural scenes right now and talk a little bit about these behaviors. So optic flow induces stabilization behaviors. in particular the optomotor response and the optokinetic response. And so when animals, in this case, do a rotation, a counterclockwise turn, they experience this kind of visual flow field, motion moving to the right. During a forward translation, they would experience motion moving backwards, and in a receding motion, they would experience a flow field moving forward. Now, if an animal is stationary and being moved by the environment, such as a fish in a flowing river, they would be moving backward due to the current, but the world would be moving forward. And so fish have a tendency to want to follow the optic flow, presumably to reduce them being swept downstream. And so this can be studied in the lab. And so here I have a movie, about 20 years old, quite a famous old movie from Mike Orger in Hervik Bayer's lab, showing larval zebrafish in a runway. And this movie is not playing very well, unfortunately. It was playing well 10 minutes ago. But you'll have to trust me here when I say that these fish are following a moving grating to swim quite robustly with the direction of optic flow. And Mike has also showed in subsequent papers that both Fourier and non-Fourier motion detection is used by these animals. Now, hold on. Having a sharing issue. Okay, I'm back. I think. Nora, can you see my screen?
Currently not.
No.
Now I can see your entire screen, yes.
Now, okay, good. So here's this video again playing, again, not so well. So anyway, I will move on to the optokinetic response. So here, this is a video from the Ehrenberg lab showing a larval zebrafish that is head mounted. And as the video plays, optic flow is being presented from right to left. And you can see the eyes producing smooth pursuits, followed by saccades to reset the eyes and allowing them to continue pursuing the object. So head fix preparations for the OMR can be used for calcium imaging studies. And so you can either have a head fix tail free preparation, or in this case, I'll play a video from this paper in 2012, where the ventral roots are recorded from in a fish that's paralyzed with bungarotoxin. And this fish has control of the visual stimulus in a closed loop fashion, such that outputs from its motor neurons allow it to control the visual stimulus. So this type of assay was used in conjunction with whole brain calcium imaging to produce the first cellular resolution brain activity maps in any animal. And what they found were many visual responsive areas in the brain, and I'll highlight area 4 here, which denotes the pretectum. And this and other studies have found that the pretectum plays a very important role in the optomotor response and the optokinetic response. Recent research on the zebrafish OMR, so this past year there's been several papers that have found important clues about how fish are processing and responding to optic flow. In this paper from the Portuguese lab, they presented zebrafish with a variety of different gratings, with different grating sizes and spatial frequencies. And this is showing the average of all those visual stimuli at the time when a fish produced a bout. And what you can see is there's this pronounced light to dark transitional zone. And so what they concluded from this is that light to dark transitions are the key driver of optomotor behavior. The Ehrenberg lab had also a paper this year where they used a cylindrical stimulation arena where the fish was placed in the middle to try to figure out where in visual space optomotor stimuli are most effective at driving swims. And so this plot is showing different zones where optomotor stimuli were presented. And this point here is the front of the fish, and these are lateral positions. And you can see hot regions in the lower temporal field for the fish. And so these zones were found to be best at driving swims. And so this is in good agreement with what the Portuguese group had found. Ruben's lab also did a recent study where they showed that fish produce behavioral and neural responses to naturalistic third-order motion cues. And this was done using so-called glider stimuli. So instead of presenting gratings, they presented these triangular type stimuli, which contain third-order motion cues. And so fish have a very sophisticated motion processing ability. So to conclude here, we know a lot about the optomotor response. We know stimuli that drive it, brain centers that process optic flow. But one thing that's lacking is an understanding of the underwater visual statistics for where these animals live. And so this is a video showing optic flow, not optic flow, but the visual environment, again, not playing very well, of fish, a fish habitat in Canada. And what you might be able to see is that in addition to the complexities of the rocks and the vegetation that is around, there's also these rippling effects that occur. And these are called caustics in optics, and they're produced by light rays being refracted at the surface and then being reflected along the bottom of the aquatic environment. So our project overview, and again, this is a collaboration funded by the Human Frontier Science Project. It's a collaboration between the Ehrenberg Lab in Tübingen, the Cooper Lab at Berkeley, the Ginty Lab in Maryland, and my lab in Toronto. The first phase was to record natural videos in a very immersive way using a 360-degree camera. We then want to quantify visual statistics, which Emily will talk about. And then in the future, look at brain activity when fish are presented with these videos, and also look at behavioral outputs. We also have a comparative approach for this project. So we're of course using zebrafish. Because we know a lot about their optomotor behavior, plus they have all these fantastic tools for systems neuroscience. But we're also interested in this African cichlid, Statotilapia bertoni. We know a lot about their behaviors in nature. There's been decades and decades of work on these African cichlids. And one of the goals is to port some of the tools, such as calcium imaging, into this species. And I should say there's a large trend right now among zebrafish researchers looking at other types of fish to study. And the Bayer lab is looking at shell-dwelling cichlids. The Orger lab is looking at giant danio, and the Douglas, Delbene, Jutkovic, and Bass labs at Danionella. So this is a video showing young bertoni, and for those who haven't seen cichlids, you'll notice these are 4 weeks old, but they're having quite dynamic social interactions. Again, another video, sorry, not playing wonderfully. But they have different swimming patterns. They have territoriality, and quite different than zebrafish at this stage. So we're excited to start working with them. And so to begin to, so back to natural scenes, first aim of this project, we took some inspiration from Jacques Cousteau, who said the best way to observe a fish is to become a fish. And so in my first ideas for getting natural videos from the scenes of these species, a drone was an initial thought. Maybe we could tether a camera to the bottom of a drone, or maybe tether a camera or mount one on top of one of these small aquatic drones. I had a sobering conversation with Tom Baden, who said, you know, he'd been to India, and he said, the water is quite shallow. This type of thing would end up being kind of a Titanic for our project. This will not work. So we came up with another idea, and that was to use a robotic system to move a 360-degree camera around an aquatic environment. And so we wanted to get this camera as far away from the components that were driving its motion. And so the core of this system is an XYZ gantry system. Each gantry has 30 centimeters of travel. And so these are the gantries here. The camera is connected via carbon fiber booms. There's a rotational motor that sits here, so we can rotate the camera with good precision. And the whole system sits on carbon fiber tripod legs, so we can get it into awkward positions in the wild. It's all controlled via a field laptop and powered by a small generator. And we named this thing Ansel for Automated Natural Scene Exploration Laboratory. And so here is in the flesh what it looks like on one of our field trips. And a little bit about the camera we use. So we use this Insta360 One X camera. It came out just in time for our studies. It's a small camera that has two cameras, one on each side, that have a wide field of view. It has nice high resolution and goes at a nice temporal frequency, 100 frames per second. And it comes with this nifty little dive case, so we didn't have to worry about coming up with a waterproof case for it. Now, Emily and her postdoc Lania did lots of calibrations of these cameras prior to us taking them to the field. They did a spatial distortion of each camera in air and underwater, rotation and translation between the two cameras, spectral sensitivity, luminance, response, linearity, modulation transfer function. And they also developed a video processing pipeline to linearize pixel intensity values, blur to remove compression artifacts, equalize angular resolution based on spatial calibration, and then generate rectilinear images. And so here's a undistorted virtual projection on a sphere. So our plan for the field was to record videos from a diversity of habitats for each species, vegetated and non-vegetated, flowing water, still water, clear and turbid. We then wanted to use the robot to get ground truth camera trajectories in aquatic and terrestrial environments. We were doing translations of X, Y, and Z, X, Y, and X, Y, and Z at different speeds. So we did 10, 40, and 60 millimeters per second. We also did rotations of 5, 20, and 50 degrees per second, and some combinations of translation and rotation. And finally, we took stationary videos at all locations. Okay, on to the field work. So in August of last year, we went to Zambia. We went to one of these big rift lakes, which is the home to African cichlids. We went to Lake Tanganyika, the southeast corner. And we sampled from 6 sites of various diversity in habitat, river site above Colombo Falls, 2 other river sites, and 3 sites along the lake shore. In total, we got 364 videos, about 300 gigabytes of data. Just some pretty shots from Zambia. Here's where we stayed. Cichlid researchers go there at least a few times a year and have really set up a wonderful operation down there. Here's the robot and some different habitats we recorded from. There's a postdoc, Venkatesh, and grad student Nicholas operating the robot in one of the scenes, or one of the habitats, which was actually in a harbor. And I want to point out Walter Salzberger from the University of Basel, who was a tremendous help in the logistics of getting us down to Zambia and helping us find cichlids in the lake. Alex Jordan was also along from the Max Planck Institute doing his own research. And this is the village chief who we met with to get approval to do our research. So here's a movie, hopefully it plays well, of Ansel in action and the Colombo River. So you can see it's doing XYZ trajectory. You can hear in the background the generator, which is powering it. So we have all the cables running into the boat. Here's Nick running the laptop. So we typically were at a site for 2 to 3 hours with setup and doing the full set of trajectories. So here is a movie, 360 movie of an XYZ rightward trajectory at 60 millimeters per second. It's slowed down a bit. So the movie has started. And now is kind of liftoff. We're moving in this XYZ plane. And I am crudely, digitally just moving the camera around. The camera wasn't actually rotating, but just to give you a sense of the 360-degree environment. Again, I'm just panning around. And I think now I will show you just that we have a nice full view of the bottom there. So the camera has worked quite well. The robot worked quite well. So we think we have a nice set of videos from these habitats. And the robot is actually off in the distance here, which you can't see the legs due to the vegetation. This was a particularly nice site. We had the full leg extension, so we were in a couple meters of water here. And I'll just give you a movie overview of the site. Here is a reeded area very close to one of the villages. And here is the underwater scene from that location. And we saw lots of cichlids. This is a adult female Bertoni. They were often just munching on vegetation, but we probably saw up to 50 in this location. Okay, that was Africa. On to India. So we went to India in October of this year. We are certainly not the first to do this. We were lucky to have Dr. Arunachalam and his student, Dr. Vijay Kumar, along on our trip to guide us to zebrafish locations. So he has previously sampled throughout India. Dave Perici was sort of the first Western zebrafish researcher who in the 2000s sampled several locations in Western India. I forgot to mention David Edds in the 1990s, who sampled lots of zebrafish habitats in Nepal. And of course, Tom's group went to India a few years ago, West Bengal, and Friedrich Jutfeld has also been to India at sites also in West Bengal. And recently, Benjamin Djukovic has been looking at Danionella sites in Myanmar, specifically looking at acoustic behaviors in the wild. Now here's where we went in Assam. So Guwahati was our kind of home base, and we branched out from there every day to different sites, trying to get into very natural sites, forested regions. We went up to the border of Bhutan and also some lowland regions. In total, we were at 9 sites and captured 245 videos, about 400 gigabytes of video data. Some photos of the sites we went to. Rocky streams. Here's more of a rice paddy zone. This was a small stream, a little more turbid, where we recorded in the river and then did a set of terrestrial movies very close by in a vegetated region. Some of the zebrafish our collaborators caught for their studies. We saw zebrafish everywhere. Only at one site were we unable to find zebrafish. Usually within 10 minutes you found them. This is quite a large zebrafish to find in the wild. Typically they were skinny in our collaborator samplings. Here's an example of the robot doing a rotation in a flowing stream. And just another rice paddy scene. Some more scenery. This is near the Bhutan border. I'll put a little star. Neuro was along on our trip doing her own studies. Here we are setting up in a rocky location. Here I am in a lowland forest stream, a more turbid stream. And here I just wanted to show you, so this is a forest stream in some hilly region of Assam. The water looks quite clear. We found zebrafish here as well. We also often saw this species of gillyfish. So here I want to show you, this is a video of fish swimming against the current in this stream. They're in the shadows of the robot. So these fish typically try to stand still within the stream, and a fish ecologist friend has informed me that this is a common strategy for fish that just want to stay and have their mouths open and just basically collect the conveyor belt of food that is flowing down the stream. Now from that same forest stream, this is now underwater, and I'm going to show you a rotation in the stream, and you can notice it's quite turbid. So because we're looking through more water than we were from the surface, it's quite a shallow zone. We notice that turbidity can be deceiving when you just look from the top. And so here we do a rotation. The sun was quite low at this location, so that was the sun. And this rippling, this caustic, as I mentioned earlier, this effect of light being refracted by the surface and reflecting along the bottom produces these light and dark transitions. So this is going to be a video of just zebrafish swimming along a lake shore, not a lake shore, a stream shore, I should say. But you see lots of power within these ripples moving towards the shore. And so one potential hypothesis is that these ripples look a lot like the optomotor stimuli we would produce in the lab. that these could be driving fish towards, shallower waters along the sides of a stream, potentially where protozoa or prey are accumulating. So just speculation there, but I think an interesting hypothesis. We potentially caught a wild larval zebrafish or juvenile. This was a location, a rice paddy. This was sort of the micro location where Vijay, one of the Indian scientist collaborators, was able to capture this guy. And so I'll play a video. And it's not playing great, but you have to trust me. It's doing swims that look a lot like what we see in the lab for a larval zebrafish. And if you look closely at its melanophore patterning, it looked very similar. So possible that we observed a larval zebrafish, but I think in the future, more field work would be very cool to go and have a really focused effort to find larvae. Okay, back to the project overview. I talked about our recording of natural videos. Emily, in the second half, is going to talk about quantifying visual statistics from these scenes. And of course, in the future, we have these goals to look at brain activity and behavioral responses to these natural scenes. So I'll talk a little bit about our efforts towards that goal. So the Arenberg lab, Tim and Ari in Tübingen, they've developed this really cool system for presenting these natural scenes to fish while performing calcium imaging or doing behavior. So here's the imaging setup. You have a fish mounted within a sphere, glass sphere that could be frosted to allow back projection. The sphere allows for the immersion of a high numerical aperture objective for calcium imaging. And the way it works is there's a light crafter that projects the natural scenes onto a mirror that then projects the scenes onto a pyramidal mirror, splitting that image into 4 quadrants. These parts of the visual scene are then relayed through a lens and a mirror and then wrapped onto the sphere. And so here's just a ray diagram showing the optical path for the setup. And I should say that this is near completion. There's still some fine tuning that's being done, but we hope in the next coming months to be able to start imaging. This is an early video showing a camera being kind of dunked into the sphere when a grating is being projected. You'll see some artifacts that they're working on removing, but the system seems to be working quite well. And here we projected a natural scene onto the sphere. And so this is actually a white balloon stretched over the sphere to try to increase contrast of the projection. And so this is a movie from Zambia. We have a cichlid moving across the scene. And so far things are looking quite promising. Okay, the other bit of engineering is a bit of genetic engineering done by Scott Junti's lab. And so there were no GCamp expressing cichlids when we started this project, but Scott has been a pioneer of transgenic approaches in cichlids. And so he's generated a stable line that expresses GCamp 7b throughout the brain. We chose 7b because it's one of the brighter variants, and we hope to image even at later ages, but we have quite nice expression. He also generated a tyrosinase mutant. These lack all pigment, and this will, of course, be important for 2-photon calcium imaging. And I will say last night, we received our first shipment of these cichlids from Scott's lab. This was quite an effort. The Canadian-US border is problematic right now due to the pandemic, but quite happy. We have seen very happy fish in our facility right now. So this is great news, and we hope to start working with them shortly. Okay, I will now pass it on to Emily for part 2.
All right, so I'm just gonna take over the screen share. Nora, can you confirm that audio and video all seem okay?
Yeah, it all looks good.
Perfect, thank you. So thanks to everyone, and thanks to the organizers for putting together this talk. I'm going to take over the second part today. And my goal is going to be to dig a little bit deeper into the quantitative aspects of the data set that was captured in the fieldwork that Todd just described, and present some preliminary analyses. And these analyses are really just going to be focused on the habitat of the zebrafish, because that's where we've started off. And this is all still ongoing work, so I'm going to try to give a flavor of a variety of analyses that we're doing and what we expect to learn down the line from these data. Just as a reminder, zebrafish are a popular model organism in neuroscience because of their amenability to advanced imaging methods. They have two eyes that they use to sense visual information in their environment. And here's just a simple diagram highlighting some of the key anatomical features of the zebrafish eye. And these eyes are positioned laterally on the head and provide a wide field of view of the world around them. And although their view of the world is wide, it's relatively low spatial resolution, estimated around 1 cone per visual degree at the larval stage. But even at the larval stage, visual signals elicit a range of different behaviors in this organism. Now, as Todd said, understanding vision and visually guided behaviors in model organisms requires investigating the relevant neural systems and circuits, understanding the animal's perception and the animal's behavior, but also understanding the visual demands placed by the natural habitat of the animal. And understanding that habitat is important because over the course of evolution, we believe that animals have developed a set of perceptual systems that are going to be optimized to perform within their specific environment. And so motivated by that particular observation, there's a large body of prior work that's directly examined the visual statistics and patterns that are present in imagery of natural environments. Here, I'm actually just listing a relatively small slice of the great work that's been done characterizing statistical regularities in natural imagery. And this prior work has really nicely shown that animals have visual systems that are well adapted to the specific demands of their habitat. But this work has primarily been driven by insights about terrestrial environments and terrestrial animals. And by comparison, relatively less is known about the visual demands placed by natural aquatic habitats, like those where zebrafish and cichlids are native. And so that's the gap that we're seeking to contribute to filling with this project, with a particular emphasis on motion perception. So with that in mind, I want to dig into the new data set. And the first thing that we noticed is that the environments we captured in India were quite visually diverse and different from each other. So here I'm showing 4 example images from 4 different field sites. And there are a couple commonalities you can note, like the water is relatively shallow and the floor is kind of sandy. But these environments were also pretty variable in terms of the turbidity of the water, the amount of vegetation, and the relative stillness of the water where the zebrafish were found. And that diversity is consistent with prior work that's been done characterizing the range and scope of zebrafish habitats. So to examine statistical regularities that were particular to this ensemble of aquatic habitats, we also collected a comparison data set of nearby terrestrial and superaquatic sites. And so as I start digging into some analyses, I'm going to do basic comparisons of the visual properties of the aquatic habitats to the nearby terrestrial environments. And for clarity, I'm going to use this water droplet and leaf icon to indicate samples that come from aquatic and terrestrial habitats respectively. As a reminder, each sample in the raw data set is a 360-degree video captured at 100 Hertz. And the camera is either held stationary for the duration of the capture, or the camera is rotating or translating or both in some predetermined trajectory. For the first analysis that I want to show, we started by subsampling from the 360 videos in space and time to measure some basic visual statistics. So here we took samples that were 75 degrees by 75 degrees, and that lasted for 10 seconds. And then we just selected the green channel of the video, and we linearized the pixel values, and we removed the lens distortion. And we selected this green channel because it has pretty high sensitivity near the zebrafish long wavelength sensitive cone, which is known to be important for driving behavioral responses to visual motion. This sampling resulted in 34 terrestrial samples and 43 aquatic video samples. And again, we'll initially be looking just at those captured with the stationary camera. So the first result that I want to show you is to look at just the basic histogram of light intensity levels in these samples. And I'll start with showing the terrestrial sample. In these plots, on the x-axis, we show the normalized light intensity, which is normalized by divisively normalizing the average intensity of each clip, such that values of 1 are equal to the mean of the clip, greater than 1 are brighter, and less than 1 are darker. And then on the y-axis is the probability density. So here now each line, each thin line represents an individual sample, and then the thick line indicates the average across all of the terrestrial samples. And we observed that the average distribution of light levels in the terrestrial habitats tended to be unimodal and had a pretty heavy tail in the positive direction, which is overall consistent with prior work on natural visual statistics. And so now with this method, we can compare this histogram to the aquatic videos. And we saw that the aquatic histograms had qualitatively similar properties, but on average, the shape was quite different. And overall, these results are consistent with one prior study of aquatic environments conducted by Balboa and Jivats, which suggested that intensity distributions from individual aquatic images tend to have overall lower contrast as compared to terrestrial. But to compare these histograms more directly, I selected just one feature of the histograms that I want to talk about today, and that is the amount of skewness or asymmetry in the two luminance distributions. So skewness quantifies how lopsided a distribution is, and it's helpful for understanding luminance asymmetries in natural scenes. So for example, environments with negatively skewed histograms will have a dominance of bright points. That is, there'll be relatively more values that are greater than the mean. Environments that have zero skew are going to have a balance of points that are brighter and darker than the mean. And then environments with positively skewed histograms will have a dominance of relatively dark points in the scene. Now, a large body of prior work suggests that natural scenes tend to have positively skewed histograms, and therefore a dominance of features that are darker than the average luminance in the scene. And importantly, that bright-dark asymmetry, or that dark dominance, has been shown to be exploited by different terrestrial visual systems, ranging from flies to humans, to efficiently encode spatiotemporal contrast and motion in natural environments. And interestingly, quite recently, Yıldızoglu et al. Provided evidence for similar adaptations in the zebrafish visual system. So it's particularly timely now, I think, to investigate the prevalence and pattern of bright dark asymmetries in a typical zebrafish habitat. So what I'm going to show you here to look at that is a histogram of the skewness calculated from each sample, and then we'll compare the distributions across the terrestrial and aquatic habitats. So we found that samples from both habitats on average had significantly positive skew, so more dark points. But we also saw that the terrestrial habitats tended to be more skewed overall than the aquatic. And to get this in some units that are a little easier to think about, we next quantified this difference in terms of just the overall proportion of light levels that are below the spatiotemporal average. And this is sort of a coarse definition of dark dominance or dark contrasts. And when we looked at the data that way, we found that the average was about 71% in the terrestrial habitats and 60% in the aquatic habitats. So both significantly more than half of the points in the samples. So taken together, these two bright-dark asymmetry analyses suggest that the zebrafish habitat has some nice commonalities with terrestrial habitats, which have been more well studied in prior work. But we can also note that there are clear quantitative differences between the two. And so ideally, we want to understand whether and how these differences have implications for visual cues in the environment. So for example, ability to detect predators and food, particularly in this project as that relates to visual motion. So by way of example, if we look at a visual input like this one that I'm showing here from our data set, I'm going to start playing the video. So in that yellow circle there is some behaviorally relevant information. Specifically, there's a group of fish that's swimming by, but it appears really low contrast in this visualization because in the video, the intensity and the color of the fish is very similar to the intensity and color of the background. But you could imagine that having a visual apparatus that's well tuned to the statistical regularities of your environment might facilitate detection of even weak signals like this. And so we wanted to look at that question next. So we did this by examining luminance response nonlinearities, actually optimal luminance response nonlinearities, and how they can maximize the entropy of a neural representation of light intensity values. So that is how can we transform natural visual patterns to maximize the expected value of visual information. This is an analysis based on information theory, and I think it's best illustrated by a quick little example from photography. So if you imagine that you wanted to take a nice picture for your personal website, but you ended up capturing a visual image where a lot of the important information had relatively poor contrast, kind of like the school of fish had poor contrast in the video that I showed. If you look at the image histogram of pixel intensities, you can see that what's happened is all your intensity values fall in the middle of the possible range that's mapped out here from 0 to 255. But performing an operation called histogram equalization can help you spread the wealth of your encoder and get the most out of your image data by better matching the statistics of your input. And it turns out that a simple way to do this is to apply a pointwise nonlinear transformation to each pixel value that follows the shape of the cumulative probability of the image histogram. So for example, here, if we treat the image histogram as a discrete probability distribution of light intensities, P of X, we can calculate the cumulative that I have shown on the right. And if you apply that as a pointwise nonlinearity to each pixel in your image, it gives you a new image where every value is equally likely, and the differences in your signal you can see are much easier to detect. Now, importantly, changing this pointwise transformation even slightly can have pretty big effects on the output. So here I've just made it slightly steeper, and that results in an image that doesn't really meet our criteria for optimality very well. So to return to the problem at hand of natural vision, neural systems, we know, are often thought to employ transformations that are similar to histogram equalization in order to maximize how informative the neural response is. And in the case of the visual system, the optimal form of the response nonlinearity is going to depend importantly on the average luminance histogram that is expected across the visual environment. And this principle was used really elegantly in classic work by Laughlin, which is summarized in this figure here, where he showed that the contrast response nonlinearity of large monopolar cells in the eye of the blowfly are well matched to the optimal nonlinearity that was calculated based directly from measurements of contrast levels in their habitat. And so using the same analysis, we're going to ask whether the optimal response nonlinearity differs between aquatic and terrestrial habitats. So as a reminder here, we already have the average luminance histograms that we need for this analysis. I've showed you these already. And if we integrate those to get the optimal response nonlinearity, you can see that within the range of intensity values that we have examined, we obtain two nonlinearities that appear quite different from each other. One way to intuitively look at how meaningful these differences is to then apply these as a pointwise nonlinearity to an example scene, which I've done here. And if I play this video again, this is that same video of the school of fish swimming by. And you can see that the contrast is visually higher when we apply the transformation that was derived for the aquatic habitat in particular, versus when I apply a transformation that was optimized for terrestrial scene statistics. And intuitively, this is going to be because we've stretched out the relevant pixel values a little bit more in the relevant range in the aquatic example. And in fact, when we performed a statistical analysis, we found that each nonlinearity that we calculated was significantly better at maximizing the entropy of the output values for the appropriate environment. And so this analysis suggests that the zebrafish habitat differs sufficiently from terrestrial habitats in terms of intensity statistics to place a different set of environmental pressures on encoding variations in light level across the visual field. But importantly, we also know that these histograms are a pretty coarse measure of natural visual input because they don't tell us anything about the spatiotemporal structure of the scenes. And previous studies have done a nice job of highlighting the rich spatiotemporal features that are prominent in underwater habitats. And Todd showed some really nice examples in the earlier part of the talk, such as things like rippling, vegetation moving through the water, and flowing particulates as well. So now, next, we used a spatiotemporal power spectrum analysis to quantify whether the aquatic environments have consistently different composition of spatial and temporal frequencies. I'm not going to go into the details of how to conduct an analysis like this today because I want to wrap up with showing some cool optic flow data. But briefly, what I'm showing here is the average power spectra of terrestrial and aquatic video samples in terms of temporal frequency on the x-axis and spatial frequency on the y-axis. And our analysis suggested that one of the key differences in spatiotemporal structure across these two types of habitats is that the aquatic environments seem to have more complex temporal structure, whereas the terrestrial environments seem to have more complex spatial structure within the frequency ranges that we analyzed in this analysis. So in the spectra, for example, if you look at the aquatic videos on the left, you should note the presence of more power at higher temporal frequencies. And then if you look at the terrestrial samples on the, aquatic samples on the right, terrestrial samples on the left, you should note that there's relatively higher power at higher spatial frequencies. So this type of spectral analysis is really common in natural scene statistics, and it's useful to tell us, to give us some insights on global motion statistics and global spatiotemporal patterns. But ultimately, we're interested in reconstructing and understanding optical flow all across the visual field, because that's important for the visually guided behaviors that we're most interested in. So the last thing I want to show you are some preliminary visualizations of optic flow caused by translational and rotational self-motion of the robot that Todd constructed. So for this analysis, we're going to kind of treat the two cameras of our 360 system as being approximately similar to the two eyes of a larval zebrafish. And then we'll start exploring the optic flow patterns that might be typical of the fish's natural visual experience. So here what I've done is I've warped those two camera views into a spherical coordinate system in terms of azimuth on the x-axis and elevation on the y-axis, with the left camera and the right camera mapping to two different sides of the visual field. And I'm going to first play this clip where the camera here is translating backwards relatively slowly. So you can see some motion in the environment, but you should also hopefully be able to note that global flow pattern from the camera translation. Now what I'm showing you is we used simple spatiotemporal derivative filters to measure the local motion signals across the visual field. And at a set of sample points, I'm illustrating the motion direction as a color, shown in the color bar to the right. And then in each location, the radius of the circle indicates the speed of motion in that direction. And so you can see there's a relatively consistent pattern of opposite motion in the two cameras, which is what you would expect from translational motion. But we also see substantial variability across the visual field because not all spatiotemporal derivatives are being caused by self-motion. You can examine that variability. For example, here what I've done is I've plotted a histogram of the directions of each point of local motion in each eye. And you can see that despite this variability across the whole visual field, there is still good information about the fact that there's a backwards translation happening here, which is embodied by the tendency of the right camera direction to be opposite of the left camera direction. but we also see a good amount of variability within each camera. And so from our initial analysis, we think it's likely that reliable information about self-motion is not distributed evenly across all elevations and azimuths in the visual field. So the pattern that we see during translation, as expected, is quite distinct from the pattern of optic flow that's seen during rotational motion. And I'm going to show a rotational video here. So now the video should be rotating. And this is actually a video from one of the Africa field sites, but I thought it was a particularly nice example, so I picked it to show today. So same as with the translational video, I can overlay local motion directions and speeds. And you can see that with rotational motion, you get a relatively coherent motion across both cameras in the same direction, but you can still note a good amount of variability, particularly around where the vegetation is in the lower part of the visual field. And for example, to dig into this further, now what I've done is I'm playing the video again, and underneath it, I'm playing a video where instead of direction of speed, the intensity of each pixel is, instead of direction of motion, the intensity of each pixel is now the speed of motion. And you can see these sort of hot spots where there's faster motion than elsewhere in the visual field. And these really do correspond well to locations where there are caustics and ripples in the water, and which are violating the brightness constancy assumption associated with this type of motion estimation. So importantly, we expect that these natural optic flow fields are going to differ systematically from the fields that are created by idealized synthetic stimuli that simulate pure rotation and translational self-motion when the environment is constant and when brightness constancy is conserved. And so in ongoing research in the Arenberg lab, the team is using calcium imaging and a novel noise motion stimulus that they've created to map receptive fields of pretectal neurons in terms of their sensitivity to optic flow across the visual field. And I'm showing here just an example of the optic flow receptive field of one pretectal neuron from Ari's lab. And with the natural motion data set, we're going to be able to assess how reliable these neuronal receptive fields are at representing self-motion for natural flow fields. So I just want to wrap up with a summary of the content that both Todd and I have presented today. So we've collected a novel data set of 360 videos from both zebrafish and cichlid habitats. And these include stationary video as well as ground truth translational and rotational trajectories for the targeted study of optomotor drives and visual motion. An initial analysis of the low-level visual statistics of the zebrafish habitats suggests that they differ in their luminance and contrast and power spectra from terrestrial habitats that are more typically studied for natural visual statistics, and that these differences likely have meaningful implications for the structure and function of the visual system of aquatic animals. The Arenberg lab, Thiele lab, and Junti lab are collectively developing novel imaging and behavioral paradigms to examine these issues from a neural and behavioral perspective in the lab, along with the visual statistics. And in our ongoing work, we're examining how to optimally self-stabilize in aquatic habitats with an emphasis on distinguishing between translational and rotational self-motion, on handling conflicting motion cues that are caused by shadows and highlights and rippling, and also on interspecies differences. So thanks again to everyone on the team, and thanks to the organizers today, our funders, and to everyone for their time. Thank you.
Wonderful, thank you so much. That was really, really interesting and a great talk. It's nice to see finally the results that I saw actually being recorded in India. So it's really great to see those coming together. So we have a few questions that I'm now gonna read out loud. So our very own Tom Baden is asking, have you checked if you get very different results from the pointwise nonlinear histogram matching if you use non-green as the input, for example, red-blue channels?
Yeah, that's a great question. We have pulled the samples for the other color channels, but haven't plotted the optimal nonlinearities yet. But it's definitely in the plans, and I think it would be interesting if they did come out differently.
Yeah, absolutely. Great. Tom actually had a follow-up on that, saying that also your subsampling suggests that you took the parts of the video that shows horizon. Have you looked for dorsal or ventral visual fields as well?
Sorry, can you repeat the question?
Yes. So also your subsampling suggests that you took the parts of the video that show horizon. Have you looked the dorsal ventral visual fields as well?
Yeah, so when we do that subsampling, there are a couple of things under the hood, and one of them is that if we obtain any samples that have a large number of saturated pixels, we reject the sample because that's not going to be a good representation of the image histogram. And this is not an HDR camera. So in the upper parts of the visual field, we haven't done much sampling because you probably noticed in a couple of the examples, a lot of times the camera ends up getting saturated in that region. So right now we're focused on regions where we can eliminate any saturation. I think we'll still be able to do some elevation dependent analyses, but we've been focused right now on being able to take those large chunks of the visual field where we have good representation of the different brightness levels.
That's very interesting. George Cafetsis is asking, a great talk, thank you. Maybe I missed it, but which, if not all, aquatic landscapes you use to estimate the natural statistics? Did you separate between different types of vegetations, for example?
Right now, that's just samples from, I think Todd is at the 7 or 8 different sites. Only in India.
Yeah, so those were, we had 9 sites, and I believe, some of those sites were really turbid, so I don't think they were quite useful for your analyses, but.
Well, I think we wouldn't have eliminated anything on the basis of turbidity because that would be an important scene statistic that we would want to capture. But I think there was one that we eliminated because the camera wasn't stabilized. So that might be why we have 8 instead of 9.
Yes, there were, in stationary videos in very, fast moving streams were problematic. We would have ropes and ways to try to stabilize the whole thing. But depending on the site, it was.
Yeah, I think we were able to get samples from most of the different sites. Maybe we just missed one of them. And I haven't separated them out by site to see if the different sites have statistical differences between them. One thing that we are working on now is categorizing them on a continuum of being more or less ripply, because some of the sites had more caustics and changing shadows. And so we're working on analysis to see if we can categorize those based on features of the spatiotemporal power spectrum. But we haven't tried to separate into environments with more or less vegetation or more or less turbidity specifically.
Yeah, okay. Great. We have still time for some questions. So I have Duncan Mearns asking that great pair of talks. Does moving the camera cause drag or turbulence in the water that changes statistics of motion in the visual scene?
So I can take that one, and maybe Emily has a comment too, but yeah, so the camera was on as slender a boom of carbon fiber as we could get that would be rigid enough. We ran into issues when we did a vertical movement. So when we pulled the camera out of the water, and then the camera case would be there. So that would create rippling that would be circular and outward from that camera being pulled out. Those were the major ones. Of course, there could be slight effects at the higher speeds, but typically the caustics that were produced were dominated by the wind or different turbulences within the water. So thankfully, I think that those were minimal, except for the case when the camera was actually pulled completely out of the water. So, yeah.
Yeah.
But yeah, great question.
Great. I have one more. Timothy Lee is asking, from your field work, can you speculate why UV sensitivity would be important for zebrafish?
So I would say, I mean, based on all of the work from Tom's lab, pointing to prey capture being a sort of clear behavior that UV cones would be important for. From our field work, we weren't focused on larvae. We may have found a larvae and it was swimming near the surface where UV would be quite prominent signal. But I don't think anything specifically we can say about the role of UV in behaviors. The camera wasn't particularly sensitive to UV. We thought about bringing a UV camera that we could put underwater, but technical limitations and how to house such a thing and make such a thing, we could leave to Tom potentially. But yeah, so unfortunately, I don't think we have a whole lot of information to provide there.
Great, thank you. Great, thank you for all the questions that we got so far. So as we have posted on the YouTube chat, we have the link for this Zoom talk. So everybody is welcome to join us and have a little bit more questions and more casual talk with our with Todd and Emily. So please, everybody, you're welcome to join. And thank you for everybody who has been watching our live stream. I'm going to keep the live stream going on for a couple more minutes so that people have time to grab the Zoom link and join if they wish to do so. Thank you. And we already have people joining in.
Thanks, Nora.
Yeah, thank you, Nora.
I think I'm just going to keep asking the questions because I still have Tom asking another one that your flow field analysis suggests that for translation, the ground has the biggest motion signals, while for rotation, the whole visual field seems to contain usable motion cues. Is that consistent? And can you comment how think links to OMR, OKR dependence on visual elevation?
So I can take the first part, maybe Todd can take the second part. We do expect that to be consistent. We're still working on extracting the frames from getting a nice labeled set of translation and a labeled set of rotation, so we can do some good averaging and alignment of the different environments and see what trends are common across all of the samples in our data set. But just based simply on a geometric analysis of the presumed depth statistics of these environments, I think that observation is spot on. And I think Ari's here too, so he could also maybe respond to the second part of the question.
Yeah, I agree. I think there's also for the behavior a difference that we have seen, which is partially published. The other part not yet, but it's on bioRxiv. That basically for the optokinetic response, they look rather to the sides, like that's like where the response is strongest. Whereas for the optomotor response, there are two papers now that have shown that it's coming from the back from below. So that would actually match very nicely to the motion statistics if that's what we find. What Emily just said.
Thanks. So why from below makes a lot of sense. Why from the back? I mean, you've shown that it does that, but what's the point?
Well, one idea is, I don't know, is that you want to swim away from the from things that approach you. So it might be more important to respond to something that is coming towards you, and things that go away from you. Maybe it's less of a strong driver for behavior.
Yeah, but I would say also in Rubin's analysis, where he found this very sharp cutoff between light and dark as being the trigger. In Ari's work, they found that lower temporal field, but it's slightly different location. But I think it fits quite well with it being, very close to the eyes. Ari, I don't know if you would disagree, but
no, it's certainly very close to the eyes. Yeah.
Yeah.
That's for sure. And we have
so
only
I would say, yeah, go ahead.
As I said, we've only tested and also the Portuguese lab, motion coming from backwards, and then it's like strongest in the lower temporal field. So it would be very interesting, and we want to look at that, like how it's actually for different directions as well, to see what the organization there is, whether it's like they only care about things that are in their back, or whether it's more like also dependent on the motion direction.
Yeah.
Thanks.
And with larvae swimming, presumably at the surface mostly, although we don't know that per se, but having these cues, I mean, larvae are in potentially this much water, maybe deeper. How much of a signal are they able to get from below? And also, we don't know if larvae even are in moving water, right? This optomotor response could be something that's there and really comes into play ethologically later. It's we just don't know, right? We really don't know what it's used for.
Well, it would be amazing if they don't get swept by water movements in their early life, right? I mean, they're so tiny.
Yeah. I mean, I think they certainly would. I mean, in these rice paddy locations, as you've seen, yeah, there's wind, there's things that could push them around, but in terms of currents and things, yeah, I don't know. But I think there should be like a larvae expedition.
Yeah.
Focused on, yeah, this would be cool.
Let us know when you find them. They will come.
Okay. The time of year, the location, yeah. I mean, yeah. We need to have some kind of a light that would just illuminate them, right? Like.
Yeah.
Who else is here? Is that Joe? No.
It's Duncan.
Oh, it's Duncan. Duncan.
Hi,
Duncan.
Todd.
How are you? I thought you were Joe for a second.
No,
No. Long hair and a
yeah,
beard.
that's just like not got a haircut in a while.
Good to see you.
Good to see you, Todd.
Yeah, Miguel's here too. That's Miguel.
Oh, cool. Hey, y'all.
So how far are you from imaging those cichlids?
Oh, so we have to learn how to get